Welcome to Target USMLE's MediTargets. Today's MediTarget will be transport across cell membranes. Transport across cell membranes can be divided into passive and active transport. Passive transport can be either simple diffusion or facilitated diffusion, whereas active can be classified as primary active and secondary active transport. Secondary active transport is further classified as co-transport or symport, or counter-transport, exchange transport, or antiport. Let's see the differences between passive and active transport. Passive transport is down a concentration gradient, which means the solute moves from a region of higher concentration to a region of lower concentration, in this case from 5x to 2x. Active transport is against a concentration gradient. The solute moves from a region of lower concentration to a region of higher concentration, in this case from 2x to 5x. Secondly, passive transport does not require ATP, whereas active transport requires ATP, which is the energy currency of the cell. All types of active transport requires a carrier-mediated protein. There are three characteristics to a carrier-mediated protein transport. It is stereospecific, saturable, and there is competition for the substrates. Stereospecific means, say for example, a carrier protein would only transport D-glucose and not L-glucose. When the concentration of the solute rises, the transport of the solute across the cell directly rises, but only up to a certain point, which is called the Tmax, or the transport maximum, where all the carrier molecules are saturated. In the gut lumen, there is competition between galactose and glucose for the same carrier protein. So we have three characteristics for the carrier-mediated transport, which is S, S, and C: stereospecific, saturable, and competition happens. Now let's just look at passive transport. In simple diffusion, the solute moves down a concentration gradient. There is no carrier protein that's involved and no ATP. In facilitated diffusion, it requires the presence of a carrier protein. Again, there is no ATP involved, and the concentration, it's moving down a concentration gradient. The presence of the carrier protein makes the diffusion facilitated. In fact, it is more rapid than simple diffusion. An example would be the GLUT transporters in the adipose tissue and skeletal muscle, causing increasing uptake of glucose from the outside environment to the inside of the cells in the presence of insulin. Now let's move on to active transport. In primary active transport, the metabolic energy is derived directly in the form of ATP. You see the solute moving uphill, and it has a backpack full of ATP to move against a concentration gradient. An example would be the sodium-potassium ATPase pump. Here, 3 molecules of sodium move outside the cell to a region of higher concentration, whereas 2 molecules of potassium move inside the cell to a region of higher concentration of potassium, since you know that sodium is abundant extracellularly and potassium is abundant intracellularly. Another example would be the calcium ATPase pump in the sarcoplasmic reticulum and on the plasma membrane. Proton pump would also come under primary active transport, which is present in the parietal cell. The second type is the secondary active transport. Here, the metabolic energy is indirect. It's derived from the sodium gradient which is present across the cell membrane. If you look to the left, you see a solute going downhill, which could be the sodium gradient moving from extracellular to intracellular environment. And the energy that is released during that time is used for another molecule to move uphill. For example, if you see in the first cell, you have sodium going downhill and glucose going uphill. Now both the solutes are moving in the same direction, hence it's called as cotransport or symport. If you look on the cell to your right, you see sodium going downhill, but that energy is used for calcium to go uphill, and both the solutes are moving in opposite directions. Hence, it's called countertransport or antiport. You can also see the presence of the primary active sodium-potassium pump operating in the same cell. Now, you know the main driving force is the sodium going downhill. To have that going on, the extracellular environment should have an abundance of sodium, and who do you think is maintaining that? It is the sodium-potassium ATPase pump. Hence, for secondary active transport to work, the primary active transport should be functioning in the same cell. So if you poison the primary active pump, like in the case of using ubin or digitalis, then you also stop the secondary active transport. So, to summarize, we have passive transport, simple diffusion, and facilitated diffusion. Simple diffusion does not use the carrier protein, whereas facilitated diffusion requires a carrier protein. Both of them don't require ATP, and the solute is moving downhill, that is along a concentration gradient. Whereas to the right you have the active transport, in which in primary active transport the energy that is required is direct in the form of ATP, and it is used by the solute to move uphill. In secondary active transport, one solute moves downhill, and the energy that is derived as an indirect energy is used for the other solute to move uphill. If both solutes move in the same direction, it is called cotransport, and if they move in opposite directions, they are called countertransport. So that ends today's MediTarget. Thank you for listening.